I'm Melissa Spencer, and I'm going to talk today about muscle inflammation in chronic muscle injury, in particular DMD. So in the prior mini lecture, I talked to you about how muscle inflammation modulates muscle repair, and in fact is essential to allow muscle repair to occur properly. Well, let's look at a case study that involves Duchenne muscular dystrophy. And this is a retrospective study in which patients with Duchenne muscular dystrophy were examined who either took prednisone or did not take prednisone or who had taken prednisone in the past. So this is 3 groups of patients. And what was examined was their time to wheelchair. And this correlates very well with their survival probability. And so in this graph, what you can see is the blue line are patients that were currently on prednisone, and those patients took much longer to go to wheelchair compared to patients that were either not taking prednisone or had taken prednisone prior, but not currently taking prednisone. And prednisone is an immunosuppressant, so that means it reduces immune cells into the muscle. So in this study, what it shows is that reducing immune cells in Duchenne muscular dystrophy actually benefits the disease. So this seems counter to what we learned in the prior lecture, where immune cells actually benefit muscle repair. Now, why is that? Why is it that muscle is benefited by immune cells in the case of acute muscle injury, and it is actually detrimental to have immune cells in the case of muscular dystrophy? Well, that's what we're going to learn about in our lecture today. So let's look at the example of repetitive muscle injury. So first, what I want to point out is in acute muscle injury, there is this transient period of collagen deposition. So collagen is stabilizing the wound. Collagen is extracellular matrix material being secreted by fibroblasts between the muscle fibers. And it sits there, it's not contractile tissue, it's sort of like a fibrous material, just to stabilize the wound while the process of repair is occurring. Now, if you repeatedly damage the muscle, what you see is accumulation of collagen, and you can see in that triangle as it goes up, that's representing accumulation of collagen. And you can see it between those muscle fibers as a white, sort of a white, thick material. And that is what you see in Duchenne muscular dystrophy biopsies. You see this thick, fibrous material, even in areas between muscle fibers that look relatively healthy. And fibrosis, which is what this is, this fibrous material, is not good for muscle, especially too much of it. And it's not good because it's not contractile tissue, it doesn't serve any function, and it actually makes the muscle very stiff, and it impedes range of motion. So if you have a muscle that is, it can't extend and it can't contract by that material very well, and it starts to shorten the muscle, then that's not helpful. And that is what occurs in Duchenne muscular dystrophy. So now what does that have to do with inflammation? Well, first I want to remind you what we learned in the last lecture is that the inflammation that accompanies muscle repair has to occur in a certain order and timing. And I'm showing that to you here. You remember you have to have those early cells that come in and those promote inflammation and those clean up the wound. And then you have to have those later cells that come in and those ramp down the inflammation and those promote the repair. But what happens in Duchenne is you have a series of lesions going on. And you can think of it as a lot of different acute wounds going through degeneration and regeneration at different times, so asynchronously and at different parts of the muscle. And that's what's sort of represented to you here. So you have a wound and healing, and a little bit later you have a wound and healing. And that could feasibly affect the proper order and timing of repair. And there's an experiment that was done that emphasizes this point. And this was done by Dr. Eric Hoffman's group. And so what they did two conditions. So in the first condition shown here, so you can see the muscle cell shown in yellow, they created two wounds. And both of those wounds were created a little distance apart, and they were both created on the same day. So you can see day one, those are two lesions. And they looked at the area in between. So below the muscle cell, you see that in-between area, and that top white part is showing you the sort of the light micrograph, and below he's stained for immune cells. CD11B is a marker of immune cells. CD means a cluster of differentiation, and you can look at these different CD markers to identify different types of immune cells. So this one's called CD11B. And the immune cells are staining in green. So if you look at this micrograph, you see there's a few green cells in between the two lesions, lesion 1 and lesion 2. In the second condition, what he did is he made a lesion on day 1, but then he waited 5 days, and in the same exact distance away, he made a second lesion, but 5 days later. And what you see is in that intervening region, you now see many more immune cells. You see many more of those green CD11B positive cells. So that tells you that the inflammatory response is altered when you have asynchronous lesions undergoing degeneration and regeneration closely apart from each other. The second indication that the inflammatory infiltrate is altered and probably not properly facilitating repair in Duchenne is the evidence that we have by just looking at the composition of the inflammatory infiltrate. So if you pull out all the immune cells and you do flow cytometry and you examine what is there, and you compare a cardiotoxin-induced lesion and say, what is the composition of the inflammatory infiltrate in that condition versus what is the composition of the infiltrate in an MDX mouse? And you can see that they're different. So in a cardiotoxin condition, it's almost all myeloid cells, macrophages, neutrophils, a lot of the cells I've talked to you about in the prior lecture, eosinophils. Those are mainly granulocytes. Those are cells of the innate immune system, and I'll mention that later in the lecture. But if you look at the MDX infiltrate, you start to see many more lymphoid cells. And in fact, if you look at the breakdown of what these different lymphoid cells are, they're cells you would never see in muscle injury, like B cells and NK cells. Those don't normally appear. So not only do you see a change in the composition, the percentage of the different types, but you see weird different cell types coming in that you would not normally see in muscle repair. So that has to be influencing the muscle. And we can't really say based on these data whether it's positive or negative, but we can infer based on the studies done with prednisone that it is probably negatively influencing the muscle, because we know that immunosuppressants that reduce immune cell infiltrates benefit Duchenne patients. But that's just an assumption. So I will just take a moment to talk to you about what are these different cell types. Well, we know the immune system is broken into two main sort of types of cells. There are myeloid cells and there are lymphoid cells, and these make up the innate and adaptive immune system. They all originate from a hematopoietic stem cell precursor in the bone marrow, so that's that cell at the very top of the slide. So most of the cells that participate in acute muscle injury are myeloid cells, all those cells that are involved in innate immunity. And these are cells that are not very specific. They primarily participate in microbial resistance and wound healing. But cells of adaptive immunity, those are cells that are normally involved in specific immunity. They're cells that when they are derived initially, each cell is very unique. It has a receptor that responds to a particular antigen. And when it sees that antigen, it then expands clonally. And so when, for example, a T cell, which is a type of lymphocyte, sees its antigen, it clonally expands. It can become different types of T cells. It can become a helper T cell or a cytotoxic T cell or even a memory T cell. And those are cells that kind of become quiescent but are ready and poised to act on that particular antigen at a later date, which is the basis of vaccination. And the reason I point this out is because what was seen in dystrophic muscles is expansions of T cells, clonal T cells. And this is kind of hard to understand because why is it you have a disease with a structural protein mutation, muscles break down? Why would you have something that looks like autoimmunity? Well, I don't have the answer, but it is likely because we have a breakdown of what we call tolerance. And tolerance is where our immune system learns self from non-self. But this is going on in Duchenne, and it is most likely due to the fact that we have massive breakdown of membranes, proteins spilling out, and an alteration of the immune system. We see almost every immune cell type coming into the muscle. And at this point, it's hard to say which cell types are the most important. And that brings me to this slide, which is that what we think is the main reason that the immune system is detrimental for Duchenne. It's not that any one cell type is probably the most important. It's that you have this altered inflammatory milieu and you have inflammatory mediators acting on fibroblasts. And this is the key thing. Fibroblasts are able to respond to almost every cell of adaptive and innate immunity, and that's what this slide is demonstrating. So if you look at this slide, the top part shows all the receptors on a fibroblast and the cells of innate immunity that secrete factors that can activate fibroblasts. And the bottom part shows cells of adaptive immunity and the receptors capable of acting fibroblasts from those cells. And the key thing here is that fibroblasts secrete extracellular matrix, and that is the component of fibrosis. So this is probably the manner in which the immune cell interfaces with fibroblasts to induce fibrosis. So the best evidence for a role for the immune system in promotion of disease progression in Duchenne muscular dystrophy comes from genetic modifier studies, in which changes in the genome have been associated with changes in the phenotype of Duchenne muscular dystrophy disease progression. So 2 different genetic modifiers have been identified, both of which link back to TGF-beta, which is an immunomodulator. So I'm going to tell you about these 2 genetic modifiers now. The first one is LTBP4, and LTBP4 is a TGF-beta binding protein. LTBP4 sits in the extracellular matrix and it keeps LTBP4 inactive. So it holds on to LTBP4 until it is cleaved by a protease, a serine protease. And once it's cleaved, it releases TGF-beta, and it allows TGF-beta to bind to its receptor and turn on SMAD signaling, and that promotes fibrosis. And studies by Elizabeth McNally and her colleagues have shown that a genetic modifier in mice and in humans now correlates very strongly with disease progression. And this genetic modifier is in the LTBP4 gene. So in mice, this genetic modifier changes one region of the locus. And so what I'm showing you here by this red line is the 2 different genotypes of mice. So the 129 versus the D2 mice have one small change that differentiates the 2. So in the 129 mice that are more mild, they have a little bit of the genome that's missing in the D2. And that little bit of the genome is protective for them. That bit of the gene, that part of the allele, is when it's in the protein, it makes the LTBP4 hold on to TGF-beta. So it's less active, it's less able to be cleaved by the serine protease, and you see less TGF-beta activity, and you see less disease. Likewise, in the D2 mice, because they're missing that little bit, that's a proline-rich region of the gene, of the protein, it's more likely to be cleaved, and TGF-beta is more likely to be released, more active, and you have more severe disease. And we see a similar phenotype in human patients. A second modifier of dystrophic disease is called osteopontin. And this is an immunomodulatory protein. It's made by immune cells and many other different types of cells. And it is known to act on two aspects of the dystrophic phenotype. The first is we know that osteopontin can act directly on fibroblasts to activate fibroblasts and promote fibrosis. And the second way that osteopontin acts on the disease is it changes macrophage polarization. So it can change polarization towards a more pro-regenerative or less regenerative phenotype. And so humans have been identified with different SNPs, so this is small nucleotide polymorphisms within the promoter of the osteopontin gene called SPP1, and these alterations correlate with altered disease severity. So if you look at the graph with the green bars, you can see on the abscissa that the TT genotype, those patients have a higher grip strength, and the patients with the GG have a lower grip strength. And those patients with the GG have actually higher osteopontin. So we have a case where higher osteopontin expression is correlating with lower grip strength. Similarly, those patients go to wheelchair earlier. So if you look at the next graph, the patients with the GG genotype are actually going to wheelchair. Those are the red line. They're going to wheelchair earlier. So this is a second example of a genetic modifier that correlates with disease. And osteopontin is known to have a relationship with TGF-beta. It's known to induce TGF-beta in multiple cell types. And so we now have two genetic modifiers, both linking back to an immunomodulatory protein, TGF-beta. Both of these affect disease progression. So now let's review and talk about how do you think the immune system modulates fibrosis in Duchenne muscular dystrophy? Well, one way is through direct activation of fibroblasts. So we've had an example of, for example, osteopontin acting directly on fibroblasts to secrete matrix. There are many other examples of that. Many immune cells can secrete factors, and we know that fibroblasts have an array of receptors that can bind to an array of cytokines that can turn on matrix secretion. And indirectly, the immune system can affect muscle repair. And so even though that is not a direct effect on fibrosis, that's an indirect, because if muscle can repair ahead of muscle degeneration, there is less development of fibrosis in that way. So we know that the immune system can modulate muscle repair. And so that's a second manner in which the immune system can modulate fibrosis. So that summarizes our lecture today about the immune system and chronic injury. And I hope you learned how the immune system contributes to fibrosis, development of fibrosis in Duchenne muscular dystrophy. And I thank you for your attention.